Okay, so welcome back to SID2004 Bioorganic Chemistry Lecture 5. So that's when we move on to a modern, well, I like to call it as a modern peptide synthesis. So again, there's the approach is always C to N synthesis. Okay, um, again, let's recall C to N, meaning that if you are to synthesize 4 amino acid, so the C will be always be your first amino acid, and then you grow it towards the N terminal. Okay, so this is what I mean by C to N approach. Okay, so you can still do it in a solution phase, okay, using reagent that produce a volatile byproduct, for example, so that you can easily remove it by rotovapping. So instead of using a column again and again and again, you can simply remove all the byproduct by simply do rotary evaporator. So it's more of a conventional like, but then there are still issues in terms of when you rotovap it, will you remove the solvent? If you are removing the byproduct and the solvent, then the reaction will occur, but you still need to add in more and more solvents. Okay, that's one thing. That's one issue of doing a solution phase. And the second thing is that doing solution phase, you need to make sure that the peptides that are formed will not be evaporated as well. So if you are using like a very high temperature for evaporation, or a high temperature and a very low pressure, where it might promote your peptides to fly and condense in a rotary evaporator. Okay, so instead of using a solution phase synthesis, or we call it as SPS, another approach is you can do a solid phase peptide synthesis, or SPPS, that make use of a solid support to grow your peptide chain. So you have a solid, okay, this solid can be a polymer, and then you grow your peptide here. Okay, we will see, we will compare the advantage and disadvantage of both approaches of modern peptide synthesis. So again, let's go back to the basic. Say you want to synthesize a tetrapeptide with a sequence of glycine, lysine, alanine, and glycine again. So how do you do it? Since the N to C approach has issues, so you proceed with N to C to N, and you can actually do this for both SPPS and SPS synthesis. So again, well, instead of showing it in this slide, I've already mentioned it multiple times. So you synthesize it from C moving to the N. Okay, so C to N approach. So SPS works well for small peptides, and many peptides have been synthesized in this process. I'm not going to say which peptide because I'm not a fan of SPS. I'm an SPPS guy, so I'm not going to touch about it. And a large number of chemical reactions and purifications required even for a small peptide. So SPS, you need to do a lot of chemical reactions, you need to do a lot of purifications, which in this case is rotovapping. And as you know, rotovaps sometimes takes a while, right? Probably, I'm not sure whether you have been into an organic 2 lab for your second year, but yeah, it takes a while. So sometimes it takes about half an hour to just evaporate your solvent. So similarly, in this case, if you want to evaporate your byproducts, you might need to, rotovap it for a while before you can actually get a pure peptides. So although the individual yield are excellent with large peptides, the overall yield becomes so small that the SPS approach is unusable, and also requires several months or years to actually complete one synthesis. Okay, so large amount of time required, and low overall yields are due to the lag in the purification steps, again, rotovapping. So how do you quantify the yield? So you know that in any chemical reaction, it never goes to 100% completion. Even though, when you learn in lectures, you can always see that the lecturer says A plus B equals to C. But in reality, when you go into the lab, A plus B will equal to C, of course, but then it will be probably plus D, E, F, G, or probably if you don't leave it enough time for A and B to react, you will get C plus A and B. Okay, so imagine if with the two strategies that we employ, which is protecting group and activating agent, you get a coupling efficiency of 80%. Okay, always. Imagine that it's always 80% efficiency. So when you get to your third amino acid, the maximum yield that you can get is only 64%. On the fourth amino acid, you'll get 51% yield. So as you go to the 20th amino acid, the yield is only 1.4%. So you can see that there's a huge loss. Okay, so how do I calculate this? So basically, I calculate 80% times 80% times 80% times 80%, meaning that for each coupling, there's only 80% efficiency. There's only 80% of another amino acid coupling to the amino acid that you've already synthesized. So in this case, initially it's just one, doesn't matter. So you add in the first glycine, you get 80% yield, and then you add in lysine there. Now the yield is reduced to a maximum of 64%. And then when you add in the fourth amino acid, the yield is now being reduced to about 51%. So as you go and so on and so forth until Z amino acid, then the yield is very, very low. So that's why on the previous slide it says that for a large protein and peptides, so this approach is not feasible. Okay, and on top of that, the peptide must be purified at each step of the synthesis to prevent sequential unwanted reactions of leftover and whatnot. So towards the end of the day, the yield is very, very low. Okay, so take an example, insulin. Okay, it was discovered in the 90s, early 90s, and initially because the chemistry of peptides was not established at all, neither solid, neither SPPs nor SPS, or even the conventional approach is not really established. So back in the 1921 to 1969, when insulin was discovered and the diabetes disease is now well known in the community, so companies such as Eli Lilly extract and purify insulin from animal sources, for example, from pigs and cows. Pigs and cows are the main target because, especially for pigs, because you can farm them very, very easily. So if you have two pigs, with this year, next year, you probably have 20 or even 40 pigs because they breed very, very quick. So so this picture on the left hand side and the right hand side is actually showing you on the old way where they actually extracts insulin. So those are pig pancreas. Okay, so you extract insulin from the pancreas, and this one is the purification process to actually get this insulin. So that is the purest form of insulin extract. Okay, and that one is the original. So you can see there's a lot of purification process that needs to take place before insulin can be extracted and used in humans. But however, as time progresses and more people is using insulin to control the diabetes disease, there are increased number of allergic reactions to animal products. Okay, so it actually pushes. So this issue actually pushes research for an alternative insulin supply. So from here, the technology actually grows. So in 1969, when genetic technology is being discovered, um, the structure was of insulin. Oh, wait, sorry. Not yet about gene technology. Um, so in 1969, so as a chemist, when you want to do something, if you know what you are doing, or you know your end goal, then it will be easier for you to do it. So in 1969, the structure of insulin was solved by Dorothy Mary Crowfoot Huggins. Some people just call her Dorothy Huggins. After 35 years, so she actually did a research for 35 years to try and solve the structure of insulin, and that is the structure that actually she solved. Okay, and now we know that insulin has this sequence and that sequence, and from there you can actually push for a chemical synthesis, because before this nobody knows what is the structure of insulin, nobody knows what's the amino acid sequence of insulin. So once this structure was solved, it actually promotes the discovery of the primary sequence, and it pushes the chemical synthesis forward. But in the 1978, where the era of cloning or biotechnology emerges, where people can actually produce insulin very, very easily, uh, not very, very easily, but in a reasonably quick manner once the protocol has been established. So it kind of like stopped the progression of chemical synthesis of insulin. Okay, so the peptides or protein that was synthesized using the biotechnology approach is called humulin. Okay, it's a chemically identical to human insulin. It has the same function, and you can simply produce it a lot in Escherichia coli or E. coli. So bacteria grows very, very quick. So comparing a chemical synthesis which might take a week to produce something which is very big, if you establish a biotechnology system, you can simply produce it in, I don't know, 100% more yield compared to chemical synthesis in a very, very short manner. And the best thing about biotechnology is that you just feed in the bacteria with foods or amino acids, and it will do the job for you. So it's very, very easily controlled. So if you were to compare the cloning technology or biotechnology to a solution phase conventional synthesis, then you can actually see, nobody's going to use a chemical synthesis to produce insulin or even whatever proteins that we need. So not until in 1963 where solid phase peptide synthesis was born. Okay, so SPPs actually kind of like, if you imagine biotechnology and a chemical synthesis before the era of SPPs, the biotechnology technology is way beyond what a chemical synthesis can do. But with the discovery of solid phase peptide synthesis, the chemical synthesis approach are actually catching up. Okay, and I would say nowadays it's almost equal, because chemical synthesis is more flexible, so pretty much you can do whatever you want. But the biotechnological approach are limited to the host bacteria that you can use, the sequence of proteins that you can use, and so on and so forth. But we're not gonna discuss about it in very detail. Okay, so SPPS, also known as Merrifield peptide synthesis approach. So there's this paper where it was first published in 1963, where Bruce actually talks about how he managed to synthesize peptides on a solid support. Okay, so why SPPS compared to SPS? So why solid phase compared to solution phase? Because in solid phase, you do not need for a lengthy purification process between the intermediates. So as you remember, in the solution phase, once you react two amino acids together, you have a lot of byproducts that you need to either do a column chromatography, or you need to do a rotovap to actually remove it. But in solid support, imagine if your peptide is growing on a solid support on your table, for example. What is the easiest way to remove all other impurities and leave your peptides intact onto the table? Okay, so you basically just wash it, right? So you just wash it. So that's the primary advantage of SPPS. You just wash it and you remove all the impurities. And the peptide also grows directly on the solid support, and excess reagent and byproduct can simply be removed by washing with solvents. Okay, so some of you might wonder, what does the solid support means? Solid phase mean. So you know, solution phase means that you are synthesizing it in a solution, whether it's aqueous or organic solvent. But what does a solid support, solid phase mean? So although it's not 100% a solvent-free reaction, okay, because a solution phase meaning that the reaction is being done in a solution. Solid phase doesn't mean that you can do the reaction on the surface of a table, so it doesn't work that way. So it's, you still need to use a solvent to make sure the reaction occurs, but towards the end of the day, the reaction occurs, part of it, on a solid support. Okay, the synthesis is linked to a solid support. In this case, it's a polymer beads. So C to N approach, free amino acid attached to the functional groups on the solid support. So imagine this are the beads of your solid support. Okay, and on the surface of these beads, okay, and this beads is made from polymer. So the first generation is polystyrene. So it's a polystyrene polymer, and on the surface of this polystyrene, there's this linker. Okay, and this linker is a benzene ring with a chloromethyl substituent. So the structure looks almost identical to an acid chloride. So you probably know, or you probably remember, that acid chloride is very, very reactive, even though this one is not an acid chloride, but chloride is a good leaving group. So that's what's important. It's a good leaving group. So in the presence of a carboxylic acid like this, okay, the SN2 reaction, reacting with that carbon over there, and chloride will leave as a leaving group. So you can actually grow peptides on it. Okay, so first generation benzene ring with chloromethyl substituent link on a polystyrene polymer beads, also known as the Merrifield resin. So this is the first generation resin. So nowadays there's so many different types of resin. There's so many variability between the resins, but we're not going to talk about it. If you're interested, then come to my lab. Okay, so for example, again, we'll use the tetrapeptide glycine, lysine, alanine, and glycine synthesized on the solid support. So you have your solid support here, you have your linker, and you will do your synthesis from the C to N approach again. Okay, and for simplicity, I did not put any functional groups, any protecting group, or I didn't touch anything about the activating agent that you need to actually promote this reaction forward. Okay, so how do you actually do it chemically? So you have your solid support down here. Okay, and you have your amino acid main body in square green, and your Bach amino acid in red. So for simplicity, I'll just do a red circle and a square. So square means amino acid, a circle means it's a Bach protecting group. So what you can do is, first step, you can simply react the C terminal of your amide with the solid support. Okay, it works via SN2 reaction. The carbonyl group attacks the benzyl carbon on the resin, displacing the colloidal ion, as I've shown previously. So that's how the reaction goes. So you have your now you have the first coupling or first attachment of your amino acid with Bach still intact. Okay, on the solid support. So this is the first step. So once this reaction is completed, where you actually get your first amino acid attached to it with Bach, okay, and plus all your byproducts, because this resin is a solid. So what you can do is you can actually put in a column with a filter. So put your resin over here, and then you can simply wash it using solvent. So now you can remove everything and leave a clean solid support for continuous synthesis. Okay, so once you clean it up like this, then you need to remove the Bach. Okay, so removal of Bach, as mentioned previously, you can use TFA, either neat or 50% TFA in DCM. You remove Bach and you regenerate the primary amine. And again, before each step, so once you use TFA, you will remember the resin is now in a column. Okay, so you put your TFA in, leave the TFA to react for about 1 to 2 minutes, and then you drain your TFA and then you wash it using solvent again. Okay, so that's the basic. And then what you do is you pre-activate the second amino acid. Okay, so you have your first amino acid already attached to the solid support, and what you do is you have another, say, a beaker or conical flask with your second amino acid. Okay, so that one is the column where you have your resin plus first amino acid attached to it. Okay, so you pre-activate your secondary amino acid using DCC or carbodiamide, any carbodiamide reagents, and then you transfer this into your solid support, right? So then you'll get this, another attachment of your secondary second amino acid with Bach protecting group still intact. And the efficiency of this reaction can be very, very, very high, very, very high. It can get up to 99% per coupling. Okay, so to grow your peptide, you just go and simply repeat the steps. So you repeat the cycle. So you wash it, and then you remove the Bach, and then you pre-activate your amino acid again. You put it in, leave it to react, and then you wash it again, and then you remove the Bach again. So it's a repetition. So peptide synthesis on the solid support or SPPs is very, very easy to do. Well, theoretically, anyway. So from there, you can actually grow your peptide, and finally you get your peptides that you want. Okay, so again, that one is the wrong arrow. So in chemistry, you synthesize it from C terminal to N terminal. But in biology, it's different way. Biology synthesize peptides and proteins from N to C terminal. But again, it requires a specific machinery. There's proteins, ribosomes. It's called ribosomes that involve in you know synthesizing these proteins, and it uses energy, ATP, the biological machinery, the biological molecule that contains a lot of a high amount of energy, ATP. We're not going to touch about it. We're just going to focus on chemistry. Okay, so because the synthesis is repeatable, the technique can easily be automated. So that is an advantage. In contrast, in biology, the protein is synthesized from N to C terminal, as I mentioned. And once you have your final sequence, the final step is for you to extract out the peptide from the solid support. In this case, for the first generation SPPs, we use hydrogen fluoride under a very controlled condition. Okay, so you will use a neat hydrogen fluoride at a very low temperature. Normally it's less than 5 degrees Celsius. I've did one at 20 degrees Celsius, minus 20 degrees Celsius, because at a higher degree, some peptides tend to form byproducts, tend to be more reactive, that's reacting with a different peptide. So depending on your sequence, the temperature varies a lot. And then finally, once you cleave the peptide using hydrogen fluoride, you can get your crude peptides, normally in a solid form, solid powder or white solid powder normally. So depending on the sequence of your amino acid, you can get like a off yellowish color or even a very orangey color, but normally you'll get a white solid powder. Okay, just a little bit of terminology. For SPPs, we call it orthogonal chemistry, where you normally combine Bach and T third butyl chemistry, and another one we call it as FMOC and Bach orthogonal chemistry. So this one, the Bach chemistry is more on acid labile chemistry, okay, where you use TFA to deprotect the Bach, and HF to actually remove the peptide from the solid support, while FMOC and Bach chemistry is more of a base labile, where you use piperidine to remove the FMOC from the N terminal of the peptide, the amino acid, but then you use TFA to actually remove the peptide from the solid support. Okay, so I've also mentioned it there. This is just to show you about FMOC chemistry. The process is more or less similar. The only difference is the reagents that you use, the deprotecting reagents that you use, the protecting groups that is available, but otherwise the steps and whatnot are similar. Okay, so you can pause on this slide, this lecture, to look at the slides, or you can simply just continue. Okay, so the advantages of SPPs, first and foremost, is quick and seamless. So the growing peptide can be purified by washing, as I mentioned, after each step of coupling and deprotection. So you can wash the impurity very, very quickly, and as compared to SPS, where you might need to evaporate or filter to remove the impurities. Because the peptide is covalently attached to the resin, none of the peptide is actually lost in the purification steps as compared to SPS. And the second thing is, because the step in growing the peptide chain is kind of like it's a repetition. Okay, so you can always do an automation, so you can always build a system that can do this
steps again and again. So towards the end of the day, you can synthesize more than 100 amino acid in less than a week. So you can actually synthesize. I've synthesized 50 amino acid length peptide protein in 2 days. So it's not impossible nowadays to actually synthesize even bigger amino acids. So the only limitation in here is on the resin itself, which we didn't touch, so I'm not going to talk about it. Okay, so this is the automation so that I've used before, so that's why I took a picture. So that one is a machine that we use for the Bach and TBU orthogonal chemistry, while this one over here is we normally use it for FMOC and Bach orthogonal chemistry. So it's not limited. I mean, like, you can use this for FMOC as well, and you can use that one for Bach. But that's it's, you know, because the reaction is orthogonal to one another, and they kind of like complement one another, so you cannot simply mix acid and base together, because otherwise you will produce a lot of byproducts, premature cleavages and whatnot. Okay, and that one up here are all your resin and your reaction chamber. So this is where you put your solid support, your resin, and then you put in all the amino acids, reagents, and you leave it to react. Cool, right? But unfortunately, I don't have this machine here in so when I do my peptide synthesis, I always do it in a column, which unfortunately I don't have a picture here to show it to you guys. So limitation, SPPs cannot be compared to how nature works. So biological cell, bacterial cell is able to synthesize a protein containing thousands of amino acids in milliseconds. Okay, so as I mentioned, 100 amino acid in 2 days is nothing compared to thousands of amino acid in seconds. Yeah, so and protein, and also the protein complexity. Each protein has a different complexity. Thus, different protein will require post synthesis approaches to ensure the product that you will have has its function intact. Okay, so if you recall on our lecture number 3, we did mention that the different levels of protein structure has its own function. So the bigger and because the peptide SPPs is done in a linear sequence and in organic solvent. So if you also recall back in the previous lecture, organic solvent destroys or denatures proteins. So you can imagine that synthesizing peptides on a solid support, the peptide doesn't really form any secondary structure or higher. Okay, and on top of that, their sulfate bonds are missing during synthesis, so it requires a lot of optimization to ensure correct linkages. Okay, for example, when we talk about insulin, where insulin has 3 different disulfide bonds, and even though the amino acid sequence is very short, but the disulfide bonds are very specific for each cysteines. So you can't simply just mix 2 of the insulin sequences and expect that you will get a fully functioning insulin. It doesn't work that way. So there are a lot of modifications and that you can do, and I like to say to play around to actually finally get the protein or peptide of your interest. Okay, so but if the process is successful, its potential is limitless. So herein, I'm showing you a cyclic peptides. So a peptide that I've synthesized in a cyclic. So it's a cyclic. So normally, as you've learned and as I've shown you before, you always have an N terminal, you have your amino acid sequence, and then you have your C terminal. But this peptide, there's no N and there's no C terminal. Okay, so I actually cyclize the peptides. So that's what we call it as cyclic peptide. And on top of that, you can actually form a disulfide bond as shown here. Okay, so that's me in the middle there, working when I was doing my postdoctoral back in Australia. Okay, so I think that's it. I've added here the website for my group. It's not fully completed yet, but you can go and have a visit to see a little bit on peptides, and I even uploaded all these lecture notes on the website. And I think that's it. Thank you and have a good day.